Warning: the bibliographic references and the publisher's source article are accessible in the description under the video. If you want to support the dissemination of scientific knowledge, subscribe, and can you give it a thumbs up please? Abstract: we developed a new rodent model of reversible brain anoxia and performed continuous electrocorticographic and intracellular recordings of neocortical neurons to identify in real time the cellular and network dynamics that successively emerged throughout the dying-to-recovery process, along with a global decrease in electrocorticogram amplitude. Deprivation of oxygen supply resulted in an early surge of beta-gamma activities, accompanied by rhythmic membrane depolarizations and regular firing in pyramidal neurons. Electrocorticogram and intracellular signals were then dominated by low-frequency activities which progressively declined towards isoelectric levels. Cortical neurons during the isoelectric state underwent a massive membrane potential depolarizing shift, captured in the electrocorticogram as a large amplitude triphasic wave known as the wave of death. This neuronal anoxic depolarization, associated with a block of action potentials and a loss of cell integrative properties, could however be reversed if brain reoxygenation was rapidly restored within 2-3 min. The subsequent slow repolarization of neocortical neurons resulted in a second identifiable electrocorticogram wave we termed wave of resuscitation, since it inaugurated the progressive regaining of pre-anoxic synaptic and firing activities. These results demonstrate that the wave of death is not a biomarker of an irremediable death and unveil the cellular correlates of a novel electrocorticogram wave and may be predictive of a successful recovery. The identification of real-time biomarkers of onset and termination of cell anoxic insult could benefit research on interventional strategies to optimize resuscitation procedures. 1. Introduction: elucidating the neuronal events subtending the dying process during brain anoxia, the ultimate cause of death, and those occurring during the regaining of cerebral functions, while a timely resuscitation procedure is initiated, is among the most challenging issues of clinical neurology and neurosciences. Previous studies addressing this question in animal models of global brain anoxia have identified sequential changes in cortical electroencephalographic patterns during the dying process. For example, inducing an abrupt cardiac arrest in rats by intracardiac injection of potassium chloride led to a fast decline in electroencephalogram amplitude associated within the first 30 s preceding the isoelectric line, with a surge of widespread high-frequency gamma oscillations. In another cardiac arrest rodent model, wherein anoxia was induced by an interruption of the artificial ventilation, the attenuation of electroencephalogram activity towards the isoelectric state was accompanied by decreased power in all frequency bands and an increased functional connectivity between bilateral frontal cortices. Interruption of cerebral blood flow by decapitation in rats also resulted in a rapid loss of global electroencephalogram power, occasionally associated with slow waves or low amplitude fast activity. Around a minute after decapitation, during the electrocerebral silence, a slow and large amplitude polyphasic wave was suddenly detected on the electroencephalogram. This wave was suggested to mark the synchronous and irreversible death of neurons and therefore named the wave of death. Based on a biophysical single-cell model, Sant proposed that the wave of death would reflect the massive and simultaneous depolarization of a large number of neurons caused by a cascade of oxygen deprivation-gated neuronal mechanisms initiated by the dysfunction of the adenosine triphosphate-dependent sodium-potassium pumps and the loss of neural transmembrane potential. Similar observations were made from critically ill patients receiving palliative care. By means of electroencephalogram-derived multivariate indexes, a transient rise in activity, presumed to represent high-frequency brain signals, was identified within 1-2 min after cardiac arrest. This finding, suggesting the persistence in the dying brain of activity, patterns generally associated with conscious, waking state, led to the hypothesis that end-of-life surges of electrical activities could provide a neuronal basis to the near-death experiences. These vivid mental experiences, reported by many cardiac arrest survivors. A propagating polyphasic electroencephalogram wave, considered as a terminal spreading depolarization analogous to the rodent wave of death, was also recently detected during the dying process in patients with devastating brain injury after withdrawal of life-sustaining treatments. The neuronal origins of these near-death brain electrical activities recorded in vivo remain unknown, since previous human and animal studies were limited to their observation in the electroencephalogram. In addition, the lethal protocols used in rodent models have precluded an examination of the neuronal recovery process during resuscitating procedures. Here, by combining simultaneous recordings of arterial blood gases, heart rate, electrocorticographic potentials, intracortical direct current shift and neocortical neurons' intracellular activity in a rodent model of transient anoxia, we provide the first in vivo characterization of the cellular and synaptic changes subtending the electrocorticogram, dynamics that successively emerge throughout the anoxic period and subsequent resuscitation process. We also demonstrate that the anoxic depolarization of neocortical neurons causing the wave of death can be reversed and identify a new relevant electrocorticogram wave, the wave of resuscitation, which inaugurates the recovery of neuronal and network activities following a timely reoxygenation of the brain. 2. Materials and methods. Experiments were carried out in accordance with the European Union guidelines and received approval from the French Ministry for Research and the Charles Darwin Ethical Committee on Animal Experimentation. Every precaution was taken to minimize stress, suffering and the number of animals used in each series of experiments. 2. 1. Animals, surgery and assessment of physiological conditions. Adult male Sprague Doi rats N equals 29, Charles River Laboratories, weighting 300-500 g, were initially anesthetized by intraperitoneal injection of sodium pentobarbital and ketamine to perform the surgical procedures. The animals were then cannulated after incision of the trachea and placed in a stereotaxic frame. Incisions and pressure points were repeatedly infiltrated with lidocaine and the core temperature was stabilized 5 degrees Celsius throughout the experiment using a feedback-controlled heating blanket. For subsequent electrocorticogram, extracellular and intracellular recordings, small craniotomies were made above left and right primary somatosensory cortices and left primary motor cortex. After surgery, animals were paralyzed by intramuscular injection of galamine trithiodide and kept under artificial ventilation with room air at a rate of 85 cycles/min, a breath volume of 2.6 ml/cycle and an inspiratory pressure of 12 mbar. Anesthesia was then maintained during recordings by regular injection of pentobarbital. Pulse oximetry, capnography and cardiopulmonary parameters were continuously assessed with a veterinary vital signs monitor. During control periods, the arterial oxygen saturation and end-tidal carbon dioxide concentration values ranged between 95-98% and 4-5% respectively, and the heart rate was of 350-450 beats/min. Brainstem functions were evaluated by examining the pupillary state and its responsiveness to light during the different phases of the asphyxial and resuscitation procedures. A video camera, embedded into a stereo microscope, was focused on the left eye and video records were synchronized with electrocorticogram monitoring. The pupillary state was alternatively assessed in dark 1min and during illumination 2min using a 30W spotlight located at 10 cm from the eye N equals 15 protocols from 7 rats. In 4 experiments, the pupillary reflex was further tested by repeatedly shining light on the left eye. 2. Electrophysiology and data acquisition. Electrocorticogram activities were captured using silver electrodes approximately 60 k-omega, placed on the dura above primary somatosensory cortex and primary motor cortex, and a reference electrode deposed on a temporal muscle of the opposite side of the head. Electrocorticogram recordings were made from left primary somatosensory cortex 7-8 mm anterior to the interaural line, 5 mm lateral to the midline in isolation, or from bilateral primary somatosensory cortex together with the left primary motor cortex 10-11 mm anterior to the interaural line, 5 mm lateral to the midline. Electrocorticogram signals were amplified with a differential AC amplifier bandpass filtered at 0, 1 kHz and digitized at 3 kHz. Current clamp intracellular recordings were performed simultaneously with electrocorticogram monitoring using glass micropipettes filled with 2 MCAC 6070 m-omega and amplified with a 900A amplifier operating in bridge mode. Cortical neurons N equals 12 were located in the layers 5/6 of primary somatosensory cortex 1890 with 570 2 microREM below the cortical surface, Pexinos and Watson, 1986, and identified as pyramidal cells on the basis of their distinctive electrophysiological properties. Extracellular direct current potentials were obtained in primary somatosensory cortex from the vicinity approximately 300 microREM of the simultaneously intracellularly recorded cortical neurons. Direct current and intracellular signals were bandpass filtered between direct current and 3 kHz and digitized at 10 kHz. 2. 3. Asphyxial resuscitation procedures and states segmentation, physiological parameters and electrophysiological activities were continuously recorded during anoxic periods control, during interruption of oxygen supply and throughout the resuscitation process. Asphyxia was induced by a sudden interruption of artificial ventilation in the curorized animal's ventilation off. This led to a rapid collapse of end-tidal carbon dioxide concentration and a more progressive fall down of arterial oxygen saturation values towards undetectable values 20% or 10 mmHg of oxygen pressure, corresponding to the threshold sensibility of the life window veterinary monitor. After anoxia onset, electrical brain activities experienced transitional changes towards a sustained electrically silent state, defined as the isoelectric period. Ventilation was then restored to resuscitate the animal's ventilation on. This procedure initiated a progressive regaining of electrocorticogram and neuronal activities. We defined the start of the recovery period, early recovery, as the time point at which the electrocorticogram signal had recovered 95% of its full amplitude, regardless of its frequency content. Subsequent recording periods were segmented according to their relative timings to the return of oxygen supply plus 15 min, plus 30 min, plus 45 min, plus 60 min after ventilation on. Some of the animals n equals 12 were subjected to multiple asphyxial protocols, each protocol being separated from each other by a period of electrocorticogram recovery. 2. 4. Data analysis. Average membrane potential values were calculated as the mean of the distribution of subthreshold intracellular activity, and the amplitude of membrane potential fluctuations corresponded to the standard deviation of the distribution. Apparent membrane resistance was measured from voltage responses to injection of negative current pulses 0.51 Na, 100 ms duration applied at 5 Hz, and the membrane time, constant, was derived from an exponential decay fit applied to the current-induced hyperpolarizations. A mean spontaneous firing rate and mean membrane potential of cortical neurons were extracted from representative recording periods of 30-60 s, and the temporal variability of action potential discharge was assessed by calculating the interspike interval's coefficient of variation, which is less dependent on variations in average firing rates than the standard coefficient of variation. Frequency content of electrocorticogram signals was computed during the different brain states, by performing time-frequency analysis using a wavered decomposition analysis made with custom-written functions in MATLAB. The math works. An electrocorticogram signal xt was convolved with a complex Morlot's wavelet function, defined as, see equation in the original article. Wavelets were normalized and thus, see equation in the original article. The width of each wavelet function, see equation in the original article. Time frequency contents were represented as the energy of the convolved signal, see equation in the original article. To quantify the degree of statistical association and the time delay between electrocorticogram signals from different cortical regions, the nonlinear correlation coefficient, theta 210e, was calculated as a function of a time shift tau. This index quantifies the dependency of a signal yt on a signal xt tau. A piecewise linear regression curve is used to predict the signal yt given the values of xt tau. The variance of yt explained by the regression curve is called the explained variance, whereas the difference with the total variance is called the unexplained variance. Thus, the nonlinear correlation ratio quantifies the reduction of variance obtained by predicting the tall y values on the basis of the regression curve. See equations in the original article. 2. Viv. Statistics. Numerical values are given as mean more or less standard error of mean differences between groups were assessed using Kruskal-Wallis analysis of variance on ranks, paired and unpaired students' t-tests, or the non-parametric Wilcoxon-signed rank test and Mann-Whitney rank sum test were appropriate, the significance level was set at P0, 0.5, 3, results, 3, 1, time, course of electrocorticogram, changes from asphyxia onset to cortical activity recovery. Electrocorticogram activity during control periods was dominated by rhythmic waves at 4-7 Hz, as classically observed under pentabarbital anesthesia. The interruption of artificial ventilation of typically resulted in a fast decline of end-tidal carbon dioxide concentration, whereas the arterial oxygen saturation progressively attenuated until reaching undetectable values. See materials and methods. Within 20 s. Electrocorticogram signals remained unaffected for approximately 10 s before undergoing a progressive decrease in amplitude, often preceded by transient amplification. Changes in magnitude of the electrocorticogram were accompanied by modifications in its frequency content. We analyzed the temporal evolution of 4 standard frequency bands: delta 0, V4, 5 Hz, theta 4, 510 Hz, alpha medium beta 10 25 Hz and high beta gamma 25 50 Hz. In 65% of the experiments, we identified a short-lasting 9 more or less 1s from 2 to 15s, n equals 13 protocol surge of activity in the 10-25 and 25-50 Hz bands within 10-35s after interruption of the ventilation, corresponding to a mean power increase relative to the pre-anoxic period of 39 more or less 10% and 85 more or less 19%, respectively P0, 0.1 for both comparisons. All frequency bands then declined towards isoelectric levels except the delta frequency band, which displayed a late transient augmentation culminating at 2859 S42 more or less 1, 7s, n equals 20 protocols after removal of oxygen supply. This was evidenced by marked increase 22-fold larger on average, n equals 20 protocols, P0, 0.001 in the low 0, V-4, 5 Hz to high frequency merged 10-50 Hz power ratio compared to control values. The isoelectric state achieved 49 more or less 8s from 37-64s, n equals 20 protocols after asphyxia onset, was characterized by a 93-98% decrease in electrocorticogram amplitude associated with an almost complete suppression of cortical power in all frequency bands. This coincided with a flat membrane potential in the simultaneously recorded neurons, providing a direct demonstration of an electrocerebral silence. Notably, we observed that a large amplitude triphasic wave, similar to the previously described wave of death, systematically emerged from the flat electrocorticogram. The isoelectric state correlated with a drastic reduction of the heart rate, undetectable arterial oxygen saturation values and a dilation of the animal's pupil, which reached a maximal areactive mydriasis at the time of the wave of death. Restoration of artificial ventilation on 2-3, 5 min after asphyxia onset, led to a rapid raise of end-tidal carbon dioxide concentration and a slightly delayed within 30-40 s regaining of arterial oxygen saturation values. The return to normoxic physiological parameters correlated with the appearance of another polyphasic wave of relatively small amplitude in the electrocorticogram, which inaugurated the regaining of cortical activity and was thus called the wave of resuscitation. The early phase of electrocorticogram signal recovery, defined as the restoration of 95% of its full amplitude, was on average achieved approximately 11 min after brain reoxygenation and characterized by an increase in delta activity and a marked attenuation of the highest 10-50 Hz frequency components. High-frequency oscillations then progressively resumed, approximating control values about 60 min after restoration of the ventilation. Theta activity 4, 510 Hz remained relatively stable and similar to its prenoxic level throughout the resuscitating process, except for a slight decrease in the corresponding electrocorticogram. Power 45 min after reoxygenation. Resuming oxygen supply also led to a progressive constriction of the dilated pupil, which returned to normal size during the early phase of electrocorticogram recovery. By contrast, a lack of respiratory rescue irremediably led to the death of the animals, as attested by a continuously flat electrocorticogram and a cardiac arrest within 6-15 4 min after anoxia onset n equals 9 experiments, we next quantified the cortical dynamics during resuscitation by analyzing over time the coupling properties between distant cortical areas in experiments where electrocorticograms were simultaneously recorded from bilateral primary somatosensory cortex and left primary motor cortex n equals 17 protocols. The cupping index values between primary somatosensory cortex and primary motor cortex on the same hemisphere were higher than those measured from bilateral primary somatosensory cortex P0, 001 and remained similar to control values throughout the recovery period. The interaction strength between bilateral primary somatosensory cortex electrocorticogram signals was significantly increased at early recovery time and recovered pre-anoxic values 50 min after resuscitation onset. The analysis of time lags estimated from H2 suggested a bidirectional coupling between bilateral primary somatosensory cortex during all computed periods, whereas interactions between left primary somatosensory cortex and primary motor cortex were characterized by a positive delay of about 1 ms. 3. 2. Intracellular dynamics from asphyxia to cortical recovery. To elucidate the neuronal and synaptic mechanisms subtending the changes in electrocorticogram activity throughout the anoxic and resuscitating periods, we made in a subset of experiments n equals 12 conjoined recordings of left primary somatosensory cortex electrocorticogram, intracellular activity of subjacent, pyramidal neurons and extracellular direct current potential for the vicinity of the intracellular electrode. Pyramidal cells, electrophysiologically identified as regular spiking n equals 9 or intrinsic bursting n equals 3 neurons styriade, 2004, displayed in control a sustained background synaptic activity, resulting in a mean membrane potential of -69, 6 more or less 1 MVN equals 12 neurons, and a relatively low 3, 1 more or less 1, 3HZ, n equals 11 active neurons and irregular interspike intervals coefficient of variation equals 1, 0 more or less 0, 1. N equals 11 active neurons firing activity, the surge of high-frequency electrocorticogram activity that immediately followed the interruption of oxygen supply was associated in pyramidal neurons with ample rhythmic depolarizing potentials, leading to an elevated and more regular firing compared to control periods. These large membrane potential fluctuations were individually composed of small amplitude high frequency from 100 to 140 Hz events, presumably of synaptic origin, which correlated with the fast oscillations in the corresponding electrocorticogram during the subsequent low-frequency electrocorticogram pattern. Cortical neurons displayed slow composite membrane depolarizations, but progressively attenuated in amplitude and frequency, causing a marked decrease in spontaneous firing. This vanishing of intracellular activity ended into an isoelectric membrane potential deprived of any synaptic and firing events. The associated extracellular direct current potential displayed a slow positive shift, known to mirror an early anoxia-dependent increase in extracellular potassium cation. The correction of membrane potential with extracellular direct current potential values indicated an enhancement of membrane polarization, I16MV. During the isoelectric state 58-114S after ventilation arrest, cortical neurons exhibited a sudden sharp depolarization to, for more or less 0, 3 MV/S, n equals 12 neurons, that coincided with an abrupt negative deflection in the adjacent direct current potential and the manifestation of the wave of death in the electrocorticogram. Membrane potential shift resulted in an anoxic depolarization in pyramidal neurons, reaching a plateau value of -22, 0 more or less 3. 5 MV without applying the direct current correction and -3, 4 more or less 3, 4 MVN equals 12 neurons after correction, demonstrating an almost complete loss of transmembrane ion gradients. The neuronal anoxic depolarization, the associated isoelectric electrocorticogram, and the negative direct current potential shift were maintained until the restoration of oxygen supply. This provides the first in vivo description of the intracellular counterpart of the anoxia-induced direct current shift in the neocortex. Resuming ventilation resulted in a slow repolarization of cortical neurons 138S after ventilation on, which systematically correlated in time with the occurrence of the wave of resuscitation. After dropping to a maximal level of membrane polarization, cortical neurons were engaged in a recovery of their activity characterized by a progressive intensification of the synaptic drive that paralleled the return of electrocorticogram waves. The return of background synaptic activity in cortical neurons was much slower p0, 0.1 than its loss, and a few minutes 3.6, 1 min were necessary to retrieve pre-anoxic values of membrane potential fluctuations after restoration of oxygen supply. The early phase of electrocorticogram recovery was associated with a change in the firing dynamics of cortical neurons, which exhibited a significantly higher spike rate and a more bursty firing profile, driven by rhythmic synaptic depolarizations in phase with the augmented low-frequency waves in the electrocorticogram. 15 minutes after restoration of oxygen supply, cortical cells experienced a decrease in their ongoing firing rate towards pre-anoxic levels, and a restoration of their regular spiking pattern. Finally, in the two neurons that could be intracellularly recorded for duration 15 min, we found that membrane potential, standard deviation of membrane potential and spontaneous firing rate fully recovered values similar to those measured in control data not shown. 3. Integrative properties of cortical neurons during anoxia and recovery process. We next investigated how the integrative properties of neocortical neurons, i.e., their ability to process membrane currents, were affected in the course of the dyne to recovery process. We applied continuous series of negative current pulses n equals 5 neurons and measured the neuronal membrane input resistance and time constant, which respectively govern the ability of synaptic currents to displace membrane potential and the efficacy with which a cell summates synaptic events in time. As exemplified in Figure 5a, both cellular parameters started to decline in parallel after interruption of the ventilation, falling to 61.5% and 61.4% of control values at the establishment of the isoelectric membrane potential. Membrane resistance and membrane time constant values then continued to decrease and reached maximal attenuation at the apogee of the anoxic depolarization. The progressive neuronal repolarization that followed the restoration of oxygen supply coincided with incremental values of membrane resistance and membrane time constant, which fully regained their pre-anoxic levels when synaptic activity started to recover. Interestingly, we found that postsanable depolarizing response and firing could be generated at the termination of negative current pulses during the latest part of the anoxic depolarization and in the course of membrane repolarization. This indicates the regaining, during the early phases of resuscitation, of neocortical neurons' intrinsic excitability could proceed by more than 1 min the reappearance of synaptic and electrocorticogram activities. 4. Spatiotemporal properties and intracortical correlates of the wave of death and the wave of resuscitation. We further examined the spatiotemporal properties and cellular correlates of the two major electrocorticogram waveforms, the wave of death and the wave of resuscitation. We I'd and Z'd as respective cortical signatures of the beginning of and recovery from anoxic depolarization by means of multi-site electrocorticogram recordings. We found that the wave of death could be recorded at spatially separated sites in the same hemisphere left primary somatosensory cortex and primary motor cortex, as well as from homologous areas in both hemispheres, bilateral primary somatosensory cortex. The averages of the wave of deaths recorded at each cortical site demonstrated a very consistent shape, consisting of a large amplitude triphasic, initially positive, wave. Quantification performed on the whole set of protocols indicated that the wave of death latency did not significantly differ between the different cortices from 70 to 164 s after asphyxia onset, n equals 32 anoxia protocols, p equals 0.2. However, when considering only simultaneous recordings of the left primary somatosensory cortex and primary motor cortex electrocorticograms, we found that in approximately 76% of the cases the wave of death in primary somatosensory cortex preceded the wave of death in primary motor cortex, I10. 1 more or less 6SN equals 17 anoxia protocols, as illustrated in the lower part of Figure 6A1, showing simultaneous electrocorticogram, direct current potential and intracellular recordings from the left primary somatosensory cortex. The first positive deflection of the wave of death was tightly correlated with the onset of both the negative shift in direct current potential N equals 16 anoxia protocols and the plateau depolarization in neocortical neurons N equals 10 anoxia protocols. Yet, the early phase of the intracellular anoxic depolarization, measured at 20% of its full amplitude, was found to precede the corresponding electrocorticogram wave of death by 840s26, 6 more or less 3, 7s, N equals 10 anoxia protocols, P0, 01, indicating that a substantial proportion of neocortical neurons were already engaged in the anoxic insult many seconds before it could be detected as a wave of death in the electrocorticogram. We unequivocally identified the wave of resuscitation in approximately 61% 16 out of 26 resuscitation protocols of our attempts to restore brain activity after anoxia. The presence of the wave of resuscitation was systematically followed by recovery of electrocorticogram activity and the survival of the animal. By contrast, a lack of wave of resuscitation during an unsuccessful resuscitation procedure N equals 10 correlated with the animal's death in all cases. The wave of resuscitation had a less regular shape and a smaller amplitude than the wave of death, but its average waveform computed from the different recorded areas also revealed a consistent polyphasic profile, as illustrated in Figure 6b1. Small amplitude electrical noise was often superimposed on electrocorticogram records at the time of the wave of resuscitation. This residual artifactual signal was not caused by neuronal activity since the simultaneously recorded cells did not display any synaptic or firing events at that time. As for the wave of death, the mean delay of occurrence of the wave of resuscitation after restoration of the ventilation was similar in the three cortical areas. The physiological relevance of the wave of resuscitation as a marker of neuronal recovery was attested by its robust temporal links with the abrupt positive shift in direct current potential and the repolarization of cortical cells at the release of the anoxic plateau potential. Compared to the wave of death correlates, the restoration of the direct current potential and the repolarization of cortical cells after resuming oxygen supply exhibited more variable profiles and slower kinetics, possibly explaining the smaller amplitude of the wave of resuscitation. The onset of positive direct current shift and cell repolarization measured at 20% of the waveform's amplitude was found to lag the corresponding electrocorticogram wave of resuscitation +1, 38s for direct current potential, N equals 6 resuscitation protocols, P equals 0, 13, plus 642s for membrane potential, N equals 6 resuscitation protocols, P0, 05. Thus, the wave of resuscitation, which consistently preceded the restoration of pre-anoxic values of cell membrane potential and direct current potential, could provide a faithful neurophysiological measure of an ongoing successful recovery of brain activity. 4. Discussion. 4. 1. Neural mechanisms from anoxia to resuscitation. In line with findings in human patients and in previous in vivo models of brain anoxia, we found that the interruption of oxygen supply led to a rapid within 40-60s dissolution of electrocorticogram signals, which was preceded by successive surges of fast and slow activities. The flattening of cortical signal was correlated with a decrease in the amplitude and frequency of synaptic potentials in the simultaneously recorded pyramidal neurons, consistent with the reduction of excitatory and inhibitory synaptic currents observed in neocortical cells after anoxia. This dampening of synaptic activity likely resulted from the multiple anoxia-related presynaptic defects, including a reduction of presynaptic calcium cation currents, the subsequent complete abolition of synaptic activities and action potential firing in cortical neurons, including interneurons, since no inhibitory synaptic potentials could be detected in pyramidal cells, resulted in a fat-line electrocorticogram. However, this suppression of synaptic events did not cause, as theoretically expected, an increase in postsynaptic membrane resistance. This possibly resulted from an additional increase in membrane conductance due to an anoxia-induced opening of adenosine triphosphate-dependent potassium cation channels. As previously demonstrated in vitro from the same neuronal type, we recorded this increase in potassium cation currents may also participate to the net cell hyperpolarization observed at the establishment of the isoelectric state after subtraction of the extracellular direct current potential shift, the sharp membrane depolarization that inaugurated the so-called anoxic depolarization was presumably due to an acute effect of oxygen deprivation on cortical neurons. The general assumption is that the rapid depletion of adenosine triphosphate causes a failure of membrane adenosine triphosphate-dependent sodium cation/potassium cation pumps, producing a massive influx of sodium cation and efflux of potassium cation, and the correlative membrane depolarization as the membrane ion gradients are lost. The resulting depolarizing Plato potential is, in turn, likely responsible for an opening of voltage-gated calcium cation channels leading to an increase in intracellular calcium cation concentration and subsequent activation of calcium cation-dependent potassium cation channels, further increasing the membrane conductance. This canonic succession of cellular processes is supported by our results showing a strong parallelism between the anoxic depolarization and the shift in the local direct current potential, which is known to closely match the increase in extracellular potassium cation and decrease in extracellular sodium cation and calcium cation. This is also consistent with the progressive enhancement throughout the successive phases of the anoxic depolarization of neocortical neurons' membrane conductance as observed in the hippocampus during brain ischemia. Unlike previous in vitro investigations from hippocampal cells, synaptic events or action potentials were not systematically present during the rising phase of the anoxic depolarization. The various voltage dynamics of pyramidal neurons associated with the large depolarizing shifts could result from differences in the relative evolution of sodium cation and potassium cation concentration gradients following anoxia, as recently suggested by a computer modeling study. The anoxic depolarization was maintained until artificial respiration was restored and omission of respiratory rescue irremediably led to animal death, demonstrating that our asphyxia protocol engendered a progressively developing dying process in absence of active reoxygenation. Restoration of the ventilation initiated a recovery of neuronal membrane potential by resupplying the ion pumps with adenosine triphosphate, which consisted in a gradual repolarization with highly variable time course from cell to cell. This was accompanied by a retrieval of membrane resistance and neuronal intrinsic excitability, as attested by the current-induced post-anabol firing probably due to the recovery from inactivation of transient sodium cation channels. The subsequent slow recovery of synaptic drive and spontaneous firing closely matched the progressive regaining of electrocorticogram activity. This indicates that the duration from 2 to 3.5 min of the anoxic period investigated in the present study did not preclude, without prejudging of possible post-anoxic brain damages, the restoration of cellular and synaptic functions, which start to recover pre-anoxic levels approximately 6 min after reinstatement of oxygen supply. 4.2. Origin of the wave of death and the wave of resuscitation and their clinical significance. Asphyxia protocols were here conducted in rodents anesthetized with moderate doses of barbiturate, a pharmacological condition which could have impacted the results. However, the wave of death pattern we described shares common features including its triphasic shape, duration and latency of occurrence from the onset of brain anoxia, with that reported in awake rats after decapitation, thus excluding an anesthesia-dependent phenomenon. It is also analogous to the terminal spreading depolarization recorded in moribund patients undergoing a global cerebral ischemia following the withdrawal of life-sustaining therapies. Our intracellular recordings demonstrated its consistent temporal relationship with the neuronal anoxic depolarization and the correlative local negative direct current potential shift, both known to reflect the anoxia-induced loss of transmembrane ions gradients. Assuming that a substantial number of cortical cells undergo anoxic depolarization in a nearly synchronous manner, as attested by the corresponding large negative direct current potential shift, the raw electrocorticogram signal is likely to approximate the summed contributions of individual neurons and the triphasic shape of the wave of death to originate from the high-pass filtering of the resulting potential with a 0.1 Hz cutoff frequency, the collective repolarization of cortical neurons associated with a positive shift in the direct current potential after restoration of oxygen supply, likely due to the restoration of ion gradients, was reflected as a second polyphasic wave in the electrocorticogram, the wave of resuscitation. The relatively low amplitude of the wave of resuscitation was probably due to the high cell-to-cell variability in delay and time course of membrane potential recovery, resulting in smaller current flows within the cortical tissue. In most of experiments, where both primary somatosensory cortex and primary motor cortex electrocorticograms were simultaneously recorded, the wave of death first appeared in primary somatosensory cortex. This could result from the high vulnerability to hypoxia ischemia of this cortical region, which may then propagate the anoxic depolarization wave via spreading depression-like processes to distant cortical areas. Restoration of oxygen supply after the occurrence of the wave of death was followed in most cases approximately 61% by a wave of resuscitation that systematically inaugurated the recovery of neuronal and electrocorticogram activity. This suggests that the wave of death cannot be considered as a biomarker of a massive neuronal death or irremediable brain death, and thus may be more appropriately renamed wave of anoxic depolarization. However, we can conclude from the present study that the wave of death provides a reliable signature, in both rodents and humans, of a severe anoxic insult of neocortical neurons that can be readily recorded from the brain surface. To our knowledge, the present work is the first to describe the existence and the cellular correlates of a surface cortical wave predictive of a post-anoxic cortical recovery of the wave of resuscitation. This wave represents a faithful anticipatory biomarker of successful resuscitation, which can proceed by approximately 20 s the onset of membrane potential recovery in individual cortical neurons by means of appropriate electroencephalogram filtering in human patients undergoing resuscitation procedures. The identification of a wave of resuscitation or a lack of detection may potentially participate to the prognosis of a good post-anoxic outcome or to the diagnosis of an irreversible brain deterioration. 4.3. A new neuronal basis for near-death experiences. The protocols of asphyxia rapidly led to the establishment of an isoelectric state, shortly preceded by successive surges, fast and slow oscillations in the electrocorticogram. Such transient increases in brain electrical activity at near death have been previously observed in non-anesthetized rodents after sudden brain anoxia and suggested by bispectral index measurements after withdrawal of life support in human patients. The increase in high-frequency oscillations was interpreted as a possible correlate of a heightened conscious state that could account for near-death experiences. Our findings challenge this hypothesis. We found that the surge of beta-gamma electrocorticogram activities correlated in pyramidal neurons with regular firing at 6-7 driven by rhythmic membrane oscillations, which were shaped by the summation of high-frequency up to 140 Hz depolarizing and hyperpolarizing synaptic potentials. This pattern is similar to the transient burst of fast activity previously recorded in vivo in hippocampal networks during cerebral ischemia and shown to result from an oscillatory synaptic excitation tuned by gamma-aminobutyric acid and adenosine-dependent inhibition, regardless of a possible effect of anesthesia, which however does not preclude the occurrence of near-death experiences. The neuronal and synaptic activities we recorded in association with the increase in high-frequency electrocorticogram oscillations differed from that of neocortical neurons during waking state, ordinarily characterized by a sustained depolarizing synaptic drive and an irregular firing. Moreover, the duration of the high-frequency surge of few seconds, as estimated by the bispectral index score in humans, is seemingly too short to explain the richness and the temporal extent of the narratives from patients who experienced near-death experience. It is also improbable that near-death experiences occur from the transient isoelectric brain state and associated wave of death, since we demonstrated that cortical neurons had lost their excitability and were not spontaneously active during the state, and therefore unable to generate conscious experience. It is conspicuous that near-death experience reports require successful resuscitation and recovery of cortical and cognitive functions. We found that the time necessary to recover normoxic synaptic and firing patterns in cortical neurons and networks was much longer than that corresponding to anoxia-induced activity decline, making this period more suitable to subtant near-death experiences. The slow process of recovery was characterized by overwhelming 5-10 Hz oscillations in the electrocorticogram, and intracellular activities, together with persisting attenuated high-frequency patterns. Comparable atypical patterns of brain oscillations, associating a decrease in gamma activities with an increase in low-frequency pattern, have been reported in hallucinatory diseases, such as schizophrenia, It is thus plausible that the long-lasting disorganization of cortical dynamics characterized by a deficit in fast electrocorticogram activity, especially in sensory areas, be responsible for the stereotyped, hallucinatory-like, mental imagery that characterize some typical elements of near-death experiences. We thus suggest that the slow recovery of brain activities following resuscitation may represent a relevant time period to get deeper insight into the neuronal basis of near-death experiences. If conclusions. Our multiscale electrophysiological recordings combined with the monitoring of vital parameters demonstrate that brain anoxia triggers a temporally organized sequence of synaptic and firing patterns in the neocortex, which ends with the establishment of an isoelectric state deprived of neuronal activity. About 1 min after the silencing of cortical activities, neocortical neurons strongly depolarize to a membrane potential where firing is no longer observed and cell excitability is lost. We provide evidence that this state of depolarization block does not imply irreversible neuronal death, since a recovery of cell membrane polarization can be initiated if reoxygenation is rapidly reinstated. Membrane potential repolarization upon resuscitation is accompanied by a return of cell integrative properties followed by the progressive regaining of normoxic synaptic and firing activities in neurons and networks. The anoxia-induced massive depolarization of neocortical cells and its reversal upon return of oxygen supply respectively results in large amplitude positive and negative shifts of the cortical direct current potential, which can be captured as reproducible polyphasic electrocorticogram waveforms at the surface of the brain, the wave of death and the wave of resuscitation. Unlike the isoelectric electroencephalogram, which does not provide unequivocal information on neuronal excitability and on the timing of cytotoxic cellular processes after anoxia, the present findings demonstrate that the wave of death and the wave of resuscitation provide reliable and easily recordable biomarkers of the onset of and recovery from anoxia-related neuronal insult, although an equivalent of the wave of resuscitation has never been recorded from human patients so far. It is very likely that a collective recovery of membrane potential would also occur in human cortical neurons upon successful cardiopulmonary resuscitation. Therefore, the wave of death and the wave of resuscitation may help refine interventional emergency procedures, as they could be used as a real-time, pathophysiological marker of brain death initiating processes, wave of death, and as a cortical signature for positive recovery prognosis wave of resuscitation. In consequence, and ultimately, these results may also have implications for ethical and clinical debates on organ donation involving dying patients.